Hello and welcome to our coffee chat focusing on the ICH M10 guidelines. I'm Ellen Williams, digital editor of Bioanalysis Zone, and I'm joined today by three experts, Sarah Underwood, Larry Elverbach, and Eric Wolf, to discuss the uptake of the ICH M10 guidance, as well as the disadvantages and challenges associated with its implementation. So firstly, could our speakers please introduce themselves? So should we start with you, Sarah?
All right. Hi, I'm Sarah Underwood. I am the pharmacokinetic analyst and lead technical writer here at Alturas Analytics. I've been in the biotech and CRO industry now for 24 years and have the responsibility here of generating all of the bioanalysis reports that come from our laboratory. So I have a lot of experience in making sure that the regulatory requirements are met in our report writing and throughout the validation and sample analysis process.
Yeah, hi, I'm Eric Wolf. I'm a scientific associate vice president within the regulated bioanalytical group of MSD in West Point, Pennsylvania. I've been in the field of regulated bioanalysis for over 30 years. I review reports and review submissions coming out of our laboratory for marketing approval and other regulatory related issues.
My name is Larry Elverbach. I'm president and founder of LabIntegrity. We have a product called StudyDoc, which allows environments to help automate the generation of their GLP final reports. This product and its current version has been in place for about 5 years. I've been doing software like this for the past 20 years.
Perfect. Thank you all for those introductions. So let's get straight into the topic of discussion for today. So firstly, Sarah, could you give me a sort of brief overview of what the ICH M10 guidance is and why it's beneficial to have a collective guidance document?
Sure. So basically, this guidance mirrors a lot of what we've been using in the past. It just is that we've got more governing bodies that are adopting the same guidance document. So its purpose is for all the recommendations for guidance in bioanalytical method validation, and then for reporting of samples that are generated based on those validated methods. So it is mainly geared for pharmacokinetic and toxicokinetic data from both IND enabling non-clinical and then all the way through all the clinical phases of studies that might be performed. So it covers everything. It's not intended to cover the immunogenicity, and I'm really curious to see where those regulations and guidances will head in the future.
Yeah, I think it's important to focus on the fact that the ICH M10 really is focusing on human drugs under development, so GLP and clinical studies and the bioanalytical work associated with these studies. I think it's important to recognize, for example, that the US FDA 2018 guidance has an additional scope with respect to veterinary medicine and also includes some additional items with respect to biomarkers. So at least in its current incarnation, M10 is not going to totally replace the 2018 US FDA guidance.
Sarah, could you give us an idea of what's new for validation and sample analysis reports within the ICH M10 guidelines?
Absolutely. Beginning with validation reports, most of the changes just involve reporting any additional validation experiments as required for your method. The M10 also added within and between run calibration standard results table for accuracy and precision runs only. In addition, the cross-validation section talks about regression analysis, such as bland Altman plots or Deming regression. I think this is more of a requirement now than a request. For bioanalytical reports, the M10 focuses on reporting of curve and QC results for all accepted runs versus in the BMV, where it talked about passing and failing runs. Additionally, dilution QC tables might be a little bit longer because Dilution QCs now need to be run for both the high and the low dilution factors to support sample analysis. Also added is a summary table to capture the total number of samples that were reanalyzed and all the associated reasons for reanalysis. Lastly, the M10 delineates requirements for BA and BE studies. Basically, it looks like you need to include everything: passing, failing, chromatograms, report all the data.
Do you think we've started to see improvements already in consistency across the bioanalytical field as a result of the guidance?
Well, I don't know so much consistency. I think the issue is that most multinational pharma companies as well as CROs have tailored their workflows and tailored their processes to be applicable irregardless of where work is going to be done. So I don't think it's led too much consistency there. What I think it will do going forward, instead of having to watch out for new documents coming out of various regulatory agencies around the world, now that we have one ICH document, we don't need to worry about, for example, missing an update coming out of one of the ICH member countries with respect to bioanalytical work, which could have been an issue in the past.
Do you expect that the adoption of this guideline or these guidelines will be a smooth transition, or do you think certain laboratories or certain areas within the bioanalytical field will find this adoption difficult?
Having worked across the industry, it's going to be a bit of a bumpy road for some, and others they may not find it as difficult. It just, the implementation of each and every one of these different points is going to depend on what your governing SOPs are and your validation plans are, which are going to have to be reconfigured to make sure you're following these guidances. In addition, at least on the reporting side of things, the look and feel of a report might change a little bit. And as there's no technical templates available right now for how particularly they would like this data reported, there might be inconsistencies between companies and labs as to how data actually is reported in that regard.
I think there's still some, curiosity on the part of the industry and how regulators are going to apply and what their focus on M10 will be, because I think we've seen, at least with some regulatory agencies, different agencies focus and interpret things differently. Even individual reviewers can have different interpretations of guidance. So I think how all of those pieces shake out going forward is something that the industry is watching.
How do we ensure that interpretation and implementation of those guidance is as universal as possible?
Well, I think a lot of it is just going to be listening to other people's opinions as well, and how they are interpreting it, and having a sound scientific discussion as to what the validity of each individual's interpretation is. It would be nice to have technical guidances for how to interpret things. It was very nice, at least there's a glossary included with the ICH M10, which hopefully will help define things a little bit more clearly. But everybody's going to have a slightly different opinion on certain things. And so there's some things that are going to be a hard stop, that this isn't scientifically sound, and other items might be, okay, we can work with that.
The guidelines were originally sort of designed for LC-MS and immunoassays, and perhaps not with newer techniques like cell and gene therapies in mind. So with respects to that, do you believe that we're trying to fit maybe some of these principles that were possibly designed for LC-MS and immunoassays to techniques where they might not be so applicable anymore?
I think the basic principles of validation and bioanalytical data being generated are platform independent. You need to have a certain degree of accuracy and precision with your data in order to draw conclusions or to make decisions based on the data. And I think that as new techniques move forward, it behooves us as bioanalytical scientists to make sure that we apply the appropriate rigor against the, say, basic principles included in M10 in the work that we're doing in these new techniques and new platforms that we may be applying.
Just to add on that you take what you already know and you build on that. So if you have a cell-based assay or another assay platform, you know there's going to be inherent variability with it, and you're just going to need to state up front what you think that variability is going to be as you're moving through validation, and just be aware when you go to use that data, especially if it's for pharmacokinetic modeling, that it may or may not add more error to your model under those conditions.
And one thing the regulators have emphasized, they're interested in engaging with us with discussion of new platforms and data generated. So it really behooves us as the industry not to work in a vacuum, that if you're working through a new project, using a new platform or a new methodology, getting feedback and interacting with various regulatory authorities, I think behooves you.
Yes, and getting those interactions before you start going down a path that they may not agree with. So the earlier the discussion with regulatory agencies, I think the better.
Agreed.
Perfect. Thank you both. So if we maybe start looking towards perspectives on ICH M10 as a whole and maybe some future directions, what do you think are maybe some of the disadvantages to a collective international guideline? So do you think ultimately that the guidelines might limit creativity within bioanalysis to some degree?
I don't believe so. I think creativity can be good or bad, but the guidelines do a relatively good job at anticipating any possible assay nuances. But as Eric was mentioning earlier, sometimes, you know, as these instrumentation changes and as assay expectations change, there has to be some level of things that people want to do. But I think the guidelines don't inhibit any of that type of creativity.
I'd agree that I don't think there's anything inhibitory in the guidelines. Again, we need to use good science and make sure that the data that we're generating fits its purpose and the decisions that it's driving. And that's really our responsibility as scientists to ensure things.
Yeah, I agree with Eric that it's kind of like I equate it to driving over a suspension bridge with no guardrails. I've never ever hit a guardrail on a suspension bridge, but if they weren't there, I'm sure I would fall off of it or drive right off the edge of it. So having regulations and guidances is necessary, but I think within that there's still a lot of room for creativity and to make sure that you just need to back up what you're doing with sound judgment and documentation of it.
Overall, do you think the bioanalytical community is happy with the guidance that has been brought out? Or do you think there are still some sort of areas for improvement, perhaps?
I think the bioanalytical community basically got what they expected out of M10. I think there early in the process, there was some moves to, try to streamline or fine tune the guidance and really to question and potentially exclude some of the less scientifically driven quantities within various local guidances. But I think on the whole, some of those were successful, some weren't. So I don't think there's any surprises with respect to M10 from the bioanalytical community.
Yeah, agreed. I wish we could get rid of some of the redundancies, but I think overall it streamlined things much better.
Everything is acceptable. I don't think anybody's ever happy, but everything is acceptable. People can adapt to their own companies and their own SOPs to what the guidelines are putting out.
How do you think future guidelines are going to sort of improve on ICH M10? Do you think there are any clear areas that really need improvement or forward thinking, perhaps?
There's some parts in there I think that could use a little bit more clarity as to what they mean. So I'm rather excited, waiting with anticipation on some of the Q&As that might be released coming in the future. Hopefully that will help clear up some of the potential misinterpretations that might be occurring. But I would say as a whole, like Eric was saying earlier, it's nice that we have multiple regulatory bodies that are members of ICH that have all adopted the same guidance, so that we don't have to necessarily try and keep track of changes that are occurring on one of. So I think having an overall picture and one document will simplify things going forward for us.
And I think some of the, let's call them ambiguities that Sarah just mentioned, are being discussed in various industry forums. In the US, AAPS has had open scientific discussions online. They've had conversations with regulators at their recent meeting. Similarly, a couple of weeks ago, the European Bioanalytical Forum had an all-day workshop discussing M10 and some of the questions that industry and has with respect to interpretation. So I think, the discussion has already started. And as we begin to apply M10 and see regulators' interpretation of M10, things will continue to become clearer going forward.
I'm kind of biased. I hope there's a step. This is step 5 we're at right now, reporting. I think they need to go to a step 6 to not only do reporting, but give some guidance on what your final assay tables should look like. So I hope that's just what I hope happens, that there's a stage or a step 6 that goes down to the next nitty gritty, kind of like China's NMPA. China's NMPA says this is what your tables will look like. So when their auditors look through their reports, every single report looks the same because all the tables look the same. So that's what I hope happens. I don't know if that will, but I hope it will.
No, just to Larry's last point, I do think some templating is desirable, but again, it shouldn't be unilateral on the part of regulators. There should be discussion with the industry. I think, if you look at the experience in the US with FDA coming out with their guidance document on tables for regulatory submissions in 271, that guidance kind of came out without any industry discussion whatsoever. And it kind of took a lot of us aback with respect to it just coming out without any advance warning or discussion. So I guess the bottom line is any evolution of M10 really needs to continue to be a partnership between industry and regulators, and should not be unilaterally imposed by the regulators.
Yes, I agree.
Yeah, I would agree too that having the flexibility, especially if your experiments may not necessarily follow exactly align with what the normal process might be, that you need to be able to customize your tables.
I think one thing, before we close, that people should be mindful of is different regulatory agencies who are party to ICH have different timelines for implementation of ICH guidances. So it isn't just because M10 is published by ICH and has been published by EMA and US FDA that we as an industry should assume that M10 is the law globally. It's been said in discussions, for example, that Anvisa, the Brazilian agency, is still expecting submissions to be in accordance with their resolutions until M10 is formally published as a resolution, which is a multi-step process. So folks should be mindful that it's not a flip to switch and all agencies are now abiding by M10. There are different timelines for implementation over the next few years. So it is a little bit of some lack of harmony, at least initially, that eventually will get us to that harmonized state.
Great. So I think that's a really great point for us to bring this discussion to a close. So thank you very much to our experts for joining me today and for helping me to cover what is certainly a hot topic in bioanalysis at the moment.